In this third video, we'll discuss the origin of resting membrane potentials. We'll try to understand the ionic basis of resting membrane potentials. As we saw earlier, the Nernst equation explains how the equilibrium potential for ion diffusing across a semi-permeable membrane can be predicted or determined by the concentrations of the ion on either side of the membrane. For example, if we know the concentration of an ion inside the cell and outside the cell, then we could determine the equilibrium potential for this ion by using the Nernst equation, which therefore explains the relationship between concentration of ions and the equilibrium potential. So the Nernst equation describes the origin of an electrical potential across a membrane that is permeable to a single ion. Now, can we understand the origin of resting membrane potentials in cells using this framework? Yes, we could build upon the simple framework to actually understand the origin of resting membrane potentials. Let us see how. If you remember, the cell membrane can be considered as primarily a bilipid layer, and this bilipid layer is permeable directly to hydrophobic substances like carbon dioxide, oxygen and nitrogen, and also small molecules, other small molecules like ethanol. However, larger substances like glucose and amino acids are unable to cross the membrane, and particularly charged molecules, charged ions or polar substances like say sodium or potassium are unable to cross the bilipid layer. However, the cell membrane is not just a bilipid layer. It has embedded within it several classes of proteins, and many of these proteins actually provide transport structures across the bilipid layer, either in the form of transporter proteins or in the form of ion channels, and these facilitate the passage of larger molecules and of charged particles across the cell membrane. Now, ions, because especially because they tend to be surrounded by water molecules in solution, the size of ions is actually larger than what is expected from their molecular weight, because they tend to associate with water molecules, and these hydrated ions are repelled by the hydrophobic head groups of the bilipid layer, and therefore they are unable to cross the cell membrane directly. There are different classes of transporter proteins which help in the movement of ions across the cell membrane. These include ATP powered pumps. The sodium potassium pump is a good example for that. Then of course you have the ion channels which provide aqueous pores more or less through which ions can cross the cell membrane, and then there are transporter molecules to which ions can bind on one side and then can be released or transported to the other side. Amongst these, the ion channels support the fastest rates of ion transport. Comparatively, the pumps are much slower, and even the transporter molecules, the uniports or synports, are relatively slower. Ion channels provide a pathway for ions. The hydrated ions can pass through ion channels. Though one way of understanding ion channels is as simple aqueous pores through which ions can just flow through, in reality the hydrated ions actually interact with parts of the ion channel, and then they get to cross the ion channel, and therefore they are key for ionic transport. There are two basic types of ion channels: the non-gated channels and the gated channels. The non-gated channels are always open. They may be specific to particular ions, but they are always open, and if there is a concentration gradient, ions move across the cell membrane through this non-gated channels. The gated channels have a mechanism which can prevent the movement of ions through them, and then they are said to be in a closed state. And In the open state, putative gate-like structures which block the movement of ions can open up and allow the movement of ions. So gated ion channels particularly are important to understand many of the electrical properties of cell membranes. Gated channels are of different types. You have what are called as voltage gated channels, which are essentially channels which open or close depending upon the potential difference across the membrane in the surrounding region. And then you have ligand gated channels where the presence of specific binding molecules opens or closes the gates. Then you have mechanically gated channels where the stretch of the cell membrane or shear stress in the cell membrane determines the open or closed state of ion channels. We also need to remember that the ionic composition of the fluid inside the cell differs from the fluid outside the cell. For instance, potassium is found in high concentrations in the intracellular fluid, almost about 10 to 15 times more concentrated, in fact much more concentrated than the ECF, and similarly sodium concentrations in the ICF are much lesser in the intracellular fluid compared to the extracellular fluid. So for each of these ions there is a concentration gradient. For example, for potassium from inside to outside, whereas for sodium from outside to inside. And for each ion, equilibrium potential can be predicted using Nernst equation if we know the concentration of the ion inside and outside the cells. So for potassium, for instance, equilibrium potential would be equal to about minus 90 millivolts, because potassium tends to move from inside to outside, and therefore it carries positive charges from inside to outside and leaves behind negative charges inside the cell. Therefore, membrane potential, which is defined as the potential inside minus the potential outside, would be negative for a membrane which is permeable only to potassium and to no other ions. If a membrane is permeable to sodium alone and to no other ions, then the equilibrium potential for such a membrane for potassium for sodium would be plus 60 millivolts. This is because sodium moves from outside to inside, and it carries positive charges along with it, and therefore the inside of the membrane becomes positive with respect to the outside, and therefore the resting membrane potential would be positive in this case. Similarly, one would predict that the resting membrane potential for chloride would be minus 70 millivolts. So we know that the equilibrium potential is the potential at which the ionic flux down the concentration gradient is equal to the ionic flux down the electric gradient, and we can predict this equilibrium potential for a single ion if we know the concentration of that ion across the membrane. Now the question is, can we explain resting membrane potential on the basis of the equilibrium potential for ions? Remember, so far when we discussed equilibrium potential for ions, we have assumed that the membrane is permeable only to one ion, and then the Nernst equation is useful in those scenarios. However, in a cell membrane, in a real cell, the ECF and the ICF contain multiple types of ions. Each ion has its own concentration gradient, and the membrane presumably has different permeabilities for different ions. So how do we go from a single ion scenario in which the Nernst equation is applicable to a multi-ion scenario in order to explain the resting membrane potential? Now, look at this simple representation of a semi-permeable cell membrane. This is the inside of the cell, this is the outside of the cell. The inside of the cell has large concentrations of potassium ions, smaller concentrations of potassium ions on the outside, whereas there is much more sodium in the ECF, that is outside the cell membrane, compared to inside the cell membrane in the intracellular fluid, ICF. For the time being, we'll focus on these two ions. We can ignore the other ions like chlorides and the other anions. Now, if this is a semi-permeable membrane, then how do we try to understand resting membrane potential? Now, imagine a scenario where this membrane is permeable only to potassium. It is not permeable to sodium or to chloride or any other ions. In this situation, there would be a diffusion of potassium from the ICF to the ECF, from inside to outside, because there is a concentration gradient from inside to outside. As this happens, more and more positive charges leave the cell and exit the cell, and as a result, there is negativity building up on the inside of the cell membrane and positivity on the outside aspect of the cell membrane. Hence, we have a charged membrane with negativity inside, and this continues until the resulting electrical gradient becomes equal to the pre-existing chemical gradient. Now, as a result of this, we know that the equilibrium potential will, for potassium, would tend towards around minus 90 millivolts. Therefore, for a membrane which is permeable only for potassium at rest, that is, the resting membrane potential would be equal to the equilibrium potential for potassium, which is minus 90 millivolts. However, if you add one layer of complexity to this, let us say that the membrane is permeable only to sodium, not to potassium, only to sodium. Then there will be movement of sodium from outside to inside. Positive charges move from out to in, accumulate on the inner aspect of the membrane. The outer aspect of the membrane develops negativity, and again we would have a charged membrane. However, now if this membrane is permeable only to sodium, not to potassium, then in the resting state, in the equilibrium state, the potential across the membrane would be very close to the equilibrium potential for sodium, which would be about plus 50 millivolts. So we have seen scenarios where we can imagine a membrane which is permeable only to potassium and a membrane which is permeable only to sodium. Now, in reality, cell membranes are not permeable to single ions. They are in fact permeable to many ions, and particularly if you look at the two most important ions, potassium and sodium, the cell membrane has a much greater permeability to potassium compared to sodium. So in this kind of a scenario where a cell membrane is permeable to both potassium and sodium, however, the permeability of the membrane to potassium is much greater than the permeability to sodium. If the ratio, one would assume the ratio to be 40:1 in the resting state. In this kind of a scenario, what would happen is potassium would move from inside to outside down its chemical gradient and would build up electrical gradient in the opposite direction. Sodium would move from outside to inside, and this would build its own electrical gradient, which is pointing from in to out. So you have two electrical gradients, one due to potassium and the other due to sodium, developing as these two ions move down their concentration gradients. And there would be competition between these two ions in terms of the influence on the cell membrane potential. And what would happen is the resting membrane potential would tend towards the equilibrium potential of potassium, which is minus 90 millivolts, but at the same time it would also be influenced by the equilibrium potential for sodium, which is plus 50 millivolts. But given the situation that the resting membrane is much more permeable to potassium, 40 times more permeable to potassium than to sodium, the influence of potassium would be much greater than the influence of sodium. Therefore, the resting membrane potential would be pulled more towards the equilibrium potential for potassium than towards the equilibrium potential for sodium. And hence, a membrane which is permeable to both sodium and potassium, but which is permeable much more to potassium compared to sodium, would ultimately rest at an electrical potential which is closer to the equilibrium potential of potassium rather than that of sodium. Therefore, it would be negative. It would be around minus 70 millivolts in this specific example. So, one could say that membrane potentials are determined by the ions on either side of the membrane, the driving forces for ions across the membrane, and importantly, the permeability of the membrane to these ions. In fact, this relationship of the membrane potential to the concentration of ions and the permeability of the membrane to ions is quantified and expressed as an equation which is credited to the physiologists Goldman, Hodgkin and Katz, who stated that, who suggested that the membrane potential is a weighted average of the equilibrium potentials of the different ions involved, with weightage being given to permeability. This equation looks complex, but if you look at it closely, it is actually based, or it is derived from the Nernst equation. We can recognize that this part of the equation is exactly the same as that of Nernst equation, the constants, and again we have a logarithm of a ratio. The ratio is of the product, the ratio is of the sum of the products of permeability and concentration. Permeability and concentration for each of the ions that are involved in the system. The ratio of the concentrations is something which you are familiar with from our Nernst equation. Now what is different is that these ratios are weighted by the permeability of the membrane to the ions. Therefore, the Goldman-Hodgkin-Katz equation states that the membrane potential is a weighted average of the equilibrium potentials of the different ions involved. So at rest, potassium, the membrane is, the permeability of the membrane to potassium is high. The permeability of the membrane to sodium is much lower, and to chloride is also low compared to that to potassium. So to summarize, the origin of resting membrane potentials can be attributed to three factors. One is the differences in the concentration of ions across the cell membrane. Now we know that the intracellular concentration of potassium is much greater than the extracellular concentration of potassium. Here I stands for inside and O stands for outside, whereas the concentration of sodium outside is much greater than that inside. The second point, of course, is the differential permeability of membrane for the ions. And at rest, the potassium, the permeability of the membrane to potassium is far greater than the permeability to sodium. A third factor, which is not really clear from the previous discussion, is the role for sodium-potassium pumps. Now the sodium-potassium ATPase pumps continually pump potassium out of the cell and sodium, the potassium into the cell and sodium out of the cell. Now these pumps play an important role in maintaining the ionic gradients. These pumps ensure that the concentration of sodium inside the cell is low, and therefore there is always inwardly directed concentration gradient for sodium, and similarly they ensure that potassium levels are high inside the cell compared to the ECS, and therefore maintain outwardly directed concentration gradient for potassium. So this function of the sodium potassium pump in restoring and maintaining ionic concentrations also contributes to the resting membrane potential because of the unequal nature of the exchange. As you see, 3 potassium ions are pulled into the cell by the pump for every 2 sodium ions that are extruded out. So 3 positive charges in for every 2 positive charges out. That means that there is charge separation which is attributable to the sodium potassium pump, and it is estimated that the sodium potassium pumps contribute about -4 millivolts to the resting membrane potential. Thank you.